Okay, welcome to this video on fMRI analysis using SPM 12. So once pre-processing is done, we can continue with first level or subject level analysis. And this consists of two big steps, I would say: constructing and estimating the general linear model, or GLM, and contrasts. And for this demonstration, I will use a data set by Wakeman and Hansen, which is on face perception and face recognition. And they presented basically three types of stimuli: famous faces like Julia Roberts here, unfamiliar faces like this unknown guy, which together form real faces, essentially. And then there were scrambled versions of both famous and unfamiliar faces. And so these together are the scrambled faces condition. Here's the fMRI time series of the design for one subject in one run. And so green trials are famous trials, blue trials are unfamiliar, and red are scrambled. And you can see that this is an event-related design where these different trial types are basically intermixed. And then we have multiple blocks that are kind of separated by rest periods here shown in white. All right, now let's look at how to do first level analysis in SPM and MATLAB. So first, as usual, we have to add the path of our SPM 12 toolbox, where it's located on the computer. In this case, this is my path. And then we can start SPM by typing SPM fMRI, evaluating this. And then SPM 12 is going to start. And first, we want to specify our general linear model. And to do this, you click on Specify First Level. Then the batch editor will open again. And the first thing we have to do is specify a directory, which is kind of the output directory of our first level analysis. And so for this, I typically create a new folder in our data structure. For example, a big folder like derivatives, where we put in all kinds of analysis outputs. And then we can insert a new folder here, maybe calling it first level. And then I would create a subfolder for the current subject, sub 01. And this could be the output folder, which we specify here. So we paste this in here, select it using the dot, and this is our output directory. Next, we have to put in some timing parameters of the design. So we want to specify our design here in seconds, not in fMRI scans. The interscan interval is the repetition time, the TR of our design. This was 2 seconds for the current study. Then the microtime resolution, it says here, is the number of time bins per scan used for building regressors. And if you did slice timing correction, then this should be the number of slices. And so as I performed slice timing correction here, I will put in the number of slices, which was 33. And then the microtime onset, if you perform slice timing correction, should be the reference slice used there. And I used the middle slice, so this would be slice 17. Then under data and design, this is where we want to define our experimental design. So we basically have to tell SPM which regressors it should create and how. So when every stimulus was presented and for how long. And so for this, we click on New Subject Session. Then under Scans, this is where we enter our pre-processed fMRI data of the current run. So of run 1 first. Yeah. So if we go into the func folder of the of subject 1, then this was the SWAU images. Yeah? S for smoothed, W for warped or normalized, A for slice timing corrected for some reason, and then U for unwarped, which means corrected for motion and distortion artifacts. And these are the end images that we want to use. So in this case, run 1 of the current subject. So we go into this folder here first, paste it into the directory. Then we can filter for these files by putting in this roof symbol and then SWAU dot star, so end with something else. And then we can see it only finds these images, but now we find them for all runs. So we also have to add here run 01 again dot star. And then we just get the image for run 1. And now again, we have to be careful, as with pre-processing. We don't want to just select the first image, but all images of the time series, right? And for this, I'm going to use the trick again to put in inf here, and then it's going to show us all 208 images of this time series. Then we can simply click write, select all, and now we have all 208 pre-processed images selected, and we can press done. Then under conditions, this is where we want to specify our experimental task regressors, basically. So we click here on new condition. If you use the bits data structure, then the design is typically specified in an associated events.tsv file. So we will search for the respective file. This would be this one here, subject 1, run 1, events.tsv. And we can open this file in MATLAB by going to this folder. And then going to home, import data. We have to recognize all kinds of data files. And then it was subject 01, run 1, events.tsv. And now we can import this file. I will call it events here. And the data types are recognized correctly. So we can then simply click on import selection. And now we have this file in our workspace, so we can double click on it. And so you can see we have all kinds of trials for the different conditions, famous, unfamiliar, and scrambled, with their onsets, durations, and also other kind of information like which button was pressed by the participant or what was his or her response time. But what we mainly care about is the stimulus type, the onset, and the durations of the stimuli. So one trick I like to use here is simply to click on stimulus type and then sort, for example, ascending. And then we will see, okay, we get all the trials for the famous condition first. So we can simply enter under name, famous. Then for the onsets, you simply want to copy all of the onsets for the famous condition, copy and paste this in here. 31 trials. And for the durations, we want to do the same. Copy all of these and put them in here. And that's it. In this study, we don't have time or parametric modulations that we need to care about. And we're going to repeat the same procedure for the other types of conditions. So we're going to click on conditions again, new condition, and then enter this information for the scrambled and the unfamiliar. Maybe unfamiliar makes more sense in the order. So we continue with this. Put in unfamiliar, and then we will copy all of the onsets. Paste them in here. And also the durations. Paste them in here. And one more time, conditions, new condition. Name is scrambled. And then we will copy all of the onsets for scrambled. Paste. And then also the durations. Copy and paste. All right, and that's it. Okay, next what you want to do is put in multiple regressors. This is your opportunity to define additional regressors that you want to have on top of your experimental conditions. And typically these are nuisance regressors that you want to remove from the time series. What you should at least do here is add the 6 motion parameters that you get from realignment from the pre-processing step. So this will be the 3 translations in X, Y, and Z direction, and the 3 rotations, pitch, roll, and yaw. And this information is saved in text files in your functional folder from the pre-processing step. And these will be called RP something txt. So RP stands for realignment parameters. And so we would, for the first run of the first subject, we select this txt file. Go to multiple regressors, specify, and then this func folder is already selected, and we can search for RP something and then run 01 bold.txt, and there it is. So we can click on this, click done, and now we have added these motion parameters as additional regressors. Now you want to repeat this procedure for all of the runs of the current subject. So you would go to data and design again, and then maybe click on replicate subject session. And then you get the same at the bottom here. And now we would only have to replace these types of information for run 2, run 3, and so on. So we'll just repeat this procedure for run 2 of the current subject as another example. Now for this, we go under scans again, deselect the previous ones, and now we want to again search for something starting with SWAU, and then run 02 dot something. Then we get our run 2 pre-processed images. Again, we want to enter inf, select them all. And now we need the events file of run 2. So I will clear the previous events file, import a new one, and this will be sub1 run 2 events.tsv. Again, we call this thing events, and then I press import selection. And now we can do the same again. We sort by stimulus type, and then I copy the onsets for the famous condition. And then we have defined session 2. Oh, and one thing is missing. We need to also replace our multiple regressors file by the RP file of session 2. So we deselect this and then enter here RP run 02 underscore bold.txt, and then we have the correct motion regressors file. Now you can already see that this copying and pasting gets annoying quite quickly. So I just want to show you one alternative, which also is more applicable to scripting later. So instead of putting these conditions in manually, what you can do is define so-called condition mat files. To construct these condition mat files, you will have to do a little bit of coding, but I just want to show you here how these should be structured so you can create them. So I call these systematically like sub01 run01.mat. And when you open these, you can see that we have 3 types of cell arrays in here. one called names, another one called onsets, and a third one called durations. And so the names should be exactly the names of your conditions. And then in the respective fields of onsets and durations, you define your onsets and durations. So you put in onsets, we have the onsets for the famous faces, which is a 31 by 1 double vector. Then in the second spot, we have the onsets for unfamiliar. And then in the third, we have the onsets for scrambles. And for durations, it should look the same. So you have, again, a vector of durations for the different trials of the three conditions. And then in your batch, instead of defining all these different conditions, I'm going to delete these here. We can simply put in multiple conditions this mat file. So we would use sub01 run1.mat, for example, as the multiple conditions file for run 1 of subject 1. And of course, we can repeat the same procedure for the other runs. All right, so now we have specified the design, but what is missing is we also need to estimate it. And for this, you can click on SPM, Stats, Model Estimation. And here for the SPM mat, we can use a dependency and choose the fMRI model specification file that will be created. The other defaults are fine, and then we can simply click on Run. Okay, we can have a look at our design by clicking on Review, then going to the derivatives first level folder that we chose as the output directory. Put this in here and select the spm.mat. This is the design matrix of our general linear model. So you can see that we have essentially two sessions. These are the two blocks here. And in each session, we have the three regressors for the experimental conditions: famous, unfamiliar, and scrambled. And you can see here that at some points it becomes white, and this is where a stimulus was presented of the condition. And so we expect an increased response here. And then after these three experimental conditions, we have the 6 motion regressors, R1 to R6. And we have the same for run 2 here, famous, unfamiliar, scrambled, and the 6 motion regressors. And each run is also associated with a constant term, which models the intercept, so the average activity of the run. Okay, so now that our general linear model is set up and estimated, we can run some contrasts. And for this design, we have several contrasts of interest. First is a high-level contrast between real, so famous and unfamiliar, versus scrambled faces. This should give us brain regions involved in face perception. And then the more specific contrast between famous and unfamiliar faces should give us brain regions that are specialized for face recognition. We can also compute some more low-level contrasts, such as scrambled faces as compared to the resting baseline. This should give us brain regions that are involved in kind of basic visual perception. And then we can also run a contrast like real, so famous and unfamiliar faces versus the resting implicit baseline of the model. So this is kind of a mix between basic visual perception and face perception. But it's still interesting to do these kind of low-level contrasts to perform a sanity check. Yeah, you should expect that you get at least visual cortex here and maybe some of these face perception areas, such as the fusiform face area. Okay, to perform contrasts, we want to add to our batch here a contrast manager tab by clicking on SPM, Stats, Contrast Manager. Then you click on Contrast Manager. And again, as the SPM mat, we want to select the dependency and select the SPM mat file from the last step. And then we can click on Contrast Sessions and create a new T-contrast here. The first contrast we wanted to define is real versus scrambled faces, so the average of famous and unfamiliar faces minus scrambled faces. And so as the name, we put in real larger scrambled faces. And as the weights vector now, you need to define which regressors to add or subtract, essentially. Yeah. And so if we look at our design matrix again, then the first regressor is famous faces, the second is unfamiliar, and the third is scrambled. So we put in as the weights vector 0.5, 0.5, and -1. Yeah, so this will take half of the famous, half of the unfamiliar, so the average of the two, minus the scrambled faces regressor. We want to replicate this over the sessions, so we get the same contrast also for run 2. You only need to choose replicate and scale here if you have different numbers of runs per subject. Let's add another t-contrast by clicking on contrast sessions again and then new t-contrast. And now we want to define the contrast famous versus unfamiliar faces to identify brain regions involved in face recognition. So as the name here, we select famous, larger, unfamiliar faces. And now as the weights vector, we can see again, famous faces is regressor 1, unfamiliar regressor 2, scrambled faces not involved at all. So we put in 1, -1, 0. Yeah, so this will add famous, subtract unfamiliar, and do nothing with the scrambled. Okay, and then again, we want to replicate this over our sessions or runs. As the next t-contrast, we want to add this low-level contrast of scrambled faces versus rest. So we go to contrast sessions again, click on new t-contrast. The name is scrambled faces larger rest. And as the weights vector here, we just want to take the scrambled regressor. So we do 0, 0, famous, unfamiliar, not involved, and then 1 for scrambled. Now this is a special case of a weights vector. Typically weights vectors need to add to 0. The only exception is this, when you want to take contrast against the implicit baseline of the model, which is in our case the rest periods. Again, we want to replicate this over sessions. And then as the final contrast, we want to look at the activity for real faces versus rest. So the average of famous and unfamiliar regressors. So again, we go to contrast sessions, new t-contrast. As the name, we put in real faces versus rest. And now as the weights vector, we want to take the average of famous and unfamiliar. So 0.5, 0.5, and nothing of scrambled, 0, 0.5, 0.5, 0. I can replicate this, and then we're done. I always like to enable delete existing contrasts in case there were some in the model already. And then the final step I like to do is also show me the results. So for this, we can click on SPM, Stats, Results Report. Click on Results Report. Again, as the SPM mat, we can select the dependency, select the SPM mat file from the last step. And for contrasts, we click on New Contrast Query. And a nice little trick here is, as it says down here, if only one number is entered and this number is inf, then results are printed for all contrasts found in the SPM file. And this is what we want to do. So we put in inf, and then it will show us the results for all of our contrasts. On the subject level, I would not use any kind of multiple comparisons correction because we will do proper thresholding at the group level. So we just choose none here. As the voxel level threshold, I will use 0.001, so uncorrected. And then as the extent, I will choose 10 voxels, so we at least eliminate some very small clusters. Okay, and then we are really done. So now we can click on run again. Then it will ask us if we want to overwrite, and we do. So we click continue. And now it shows us the results. So we can cycle through our contrast by clicking on contrasts here and then change contrast. And you can go to the first contrast, real versus scrambled faces, all sessions. And you can see that we have some activations here. We can look at this on a brain template by clicking here at the bottom on Display, Overlays, and then choose Sections. And then, for example, go to Previous, choose the SPM folder, go to Canonical, Single Subject T1, and then we will overlay this on a template of the MNI brain. And you can also right-click here and click on Display, Labels, Neuromorphometrics, and then you also get some brain regions here. For example, now it shows us, okay, this is right medial frontal cortex. We also get some activation here in the precuneus, in inferior occipital regions, angular gyrus. So these are kind of areas that are associated with social processes. Also, we get here the fusiform gyrus, which might be the fusiform phase area. So this kind of makes sense that we get for real, as compared to scrambled phases, kind of the social brain. Let's look at the next contrast, famous versus unfamiliar phases. This is, of course, a quite conservative contrast, which should isolate brain activity for face recognition. So in this subject, at p less than 0.001, we actually don't get any significant activations, at least that are larger than 10 voxels big. So we could, for example, decrease the threshold here, maybe go to significance change, none, and then maybe we go to 0.01 and also no extend threshold, and then at least we get something. So go to overlays, previous sections. Again, I will enable labels, and we can see, okay, we get something here in the precentral, postcentral gyrus in both hemispheres. But of course, we should take this with a grain of salt, as this is only one subject and now the threshold is quite liberal. Then we look at the next one, contrast scrambled phases versus rest. So this, you can see, is quite liberal, but now we can also increase the threshold here. Go again to 0.001, 10 voxels, and overlay this again. And you can see now we get huge activation in the primary visual cortex, which is what we would expect. Yeah, so this is a nice sanity check that we actually get low-level visual areas when we compare scrambled phases versus rest, where subjects didn't look at anything. Okay, finally, let's look at the last contrast, real phases versus rest. Again, overlay previous sections. And this looks quite similar. Now we also get, again, strong activation of the fusiform gyrus, which might reflect the fusiform phase area. Okay, and now we're essentially done with first level analysis. So now we can also save our batch, so we can use it later by clicking in the batch editor on File, Save Batch and Script, then choose a reasonable folder, and maybe we call this something like batch first level. And then again, it will create these two files for us, this kind of script file, which calls this job file, which is actually the more interesting one for us, because this can be directly loaded again in the batch editor. And we can also use this for scripting. If, for example, we wanted to reload this, you could go to batch again, and then open load batch, load this batch first level job file, done. And now we have it back. And now we can simply replace these subject specific types of information for the next subject and run the analysis again. Okay, and now we're actually done. So thanks a lot for listening, and see you next time.